Step 3, evading host defenses. So here is a list of virulence factors from various styles of pathogen that aid pathogens in this step 3 in evading your immune system. And we'll go through them one by one. But in this list we have mycolic acid, which is part of the unique cell wall of mycobacteria, which we covered when we talked about hard to kill pathogens in class. Glycocalyx, which we've talked about extensively as well. They form the capsule and also the slime layer. Antigenic variation is something new. It has to do with evading antibodies that your body produces to fight the pathogen. M proteins is something that a very specific genus of pathogens produce to evade your defenses. Entering the host cell. So your immune system functions mainly in your bloodstream and in your lymphatic, in your lymph, so in your body fluids. If a pathogen goes into one of your cells, it can evade a lot of the elements of your immune system that's floating around in your blood. So your body fluids are what we call the extracellular fluid, the fluid that's moving through your vessels and in between tissues. So your antibodies and a lot of your blood cells are traversing your body and your body fluids, but if a pathogen enters one of your cells, it can hide from many elements of your immune system. So entering a host cell, and then enzymes. So we'll cover a bunch of enzymes that various pathogens produce to escape your immune system, your immune response. Okay. Mycolic acid. So mycolic acid, remember, is the main component of the cell wall of mycobacteria. So mycobacteria include the bacteria that cause tuberculosis and the bacteria that cause leprosy. They look a little like a fungus when they grow. They look kind of like yeast because they have this waxy look to them, and that's because mycolic acid is a wax. So it's mainly a solid lipid. So when you remember, they have this is not a membrane. This is built into the cell wall. So you have the normal peptidoglycan cell wall down here. So that's what all bacteria have. But then sprouting out from that, you have all of these other elements that are part of this mycolic acid. So the mycolic acid is the main element. You have a little bit of protein, also some other, some carbohydrates, but the main element is mycolic acid. So it's this really dense, waxy, resilient layer. It makes it's very resilient against a lot of chemicals. So when you get, again, remember, you buy antiseptic or hand sanitizer. When you buy hand sanitizer at the store, it usually says on the back if it can kill tuberculosis, because tuberculosis is resilient because of this waxy mycolic acid to most sanitizers. Another thing that mycolic acid does, aside from being resistant to chemicals, it's also resistant against phagocytosis. So you have white blood cells called phagocytes. Phagocytes, their job is phagocytosis. Phagocytosis is eating. So phagocytes, their job is to eat pathogens. They endocytose them. So phagocytosis is endocytosis of a pathogenic cell. So phagocytes are cells that traverse your body looking for invaders, and when they encounter an invader, they will phagocytize it. They will ingest it and digest it. Now, mycobacteria can be ingested by phagocytes, but this mycolic acid makes them resistant against digestion. So they have some enzymes that prevent fusion with the lysosome, so they don't even encounter, in many cases, the digestive enzymes inside the phagocytes. But also their mycolic acid is resistant against digestion, even if they do encounter the lysosome. And this is why only a minority of people who are infected with tuberculosis actually come down with the disease. So only 10% of people who are infected with the tuberculosis pathogen will come down with the tuberculosis disease. This is because for most people, the macrophages in our lungs, the phagocytes in our lungs, do a good enough job at controlling bacterial growth that there's not enough bacteria to cause disease symptoms. But the bacteria persists in healthy people's lungs indefinitely because they can't be digested by the macrophages. They're resistant against digestion. So for most people who have been exposed to tuberculosis, they have the pathogen living in them, but it's not causing them disease. And this is why if you've worked in a hospital before, you have to be tested for tuberculosis even if you're not sick, because even carriers can transmit the disease to immunocompromised individuals. Glycocalyx. So we learned earlier in the term that some bacteria form capsules around their outer membrane or form biofilms using glycocalyx. So glycocalyx is this sticky glycoprotein, and when it's loose and disorganized, it forms this oozy slime layer, which is what we call, it forms a biofilm. Right? So many bacteria can be part of this biofilm, they can all contribute to it, and you can get this really, really thick mass. And that's resilient. A biofilm is resilient against your immune system, your immune defenses, because once it becomes thick, first of all, it's a flat surface. It's really difficult for phagocytes to reach their pseudopodia around the bacteria and pick one off at a time out of that glycocalyx biofilm. So this thick mat of bacteria are stuck together in this sticky glycocalyx. The phagocytes have to be able to wrap their pseudopodia, wrap their arms around a single bacteria in order to ingest it. And it's really difficult to do that when the bacteria is part of a biofilm. In terms of the capsule, so when glycocalyx is organized and solid, it can form this capsule, sort of like a pill capsule around the bacteria. Those capsules are really, really large. So one reason they evade phagocytosis because it's actually just logistically difficult for the white blood cells to reach pseudopodia around a giant capsule. The capsules are just too big. So for a white blood cell to ingest a bacteria with a capsule, it has to pin it up against a solid surface in order for it to be able to reach its pseudopodia around to engulf it. Another reason glycocalyx in the form of a capsule resists immune cells is because it's negatively charged. So it has a negative electromagnetic charge, and you might remember that the phospholipid bilayer also has a negative electromagnetic charge. Remember the hydrophilic heads? Those hydrophilic heads are hydrophilic because they are negatively charged. Those phosphate groups are negatively charged. So your white blood cells, being animal cells, they don't have a cell wall or anything like that. Their outer layer is the phospholipid bilayer. So the phospholipid bilayer is negatively charged, the glycocalyx is negatively charged, so they actually repel each other, like two mismatched ends of a magnet repel each other. So that's another reason glycocalyx helps a bacteria evade phagocytosis. All right, why is communication among bacteria important in increasing virulence? So this is just a reminder. We already watched this talk at the beginning of the term. This is the quorum sensing talk. I'm just going to remind you that biofilms are involved with quorum sensing, and that's another reason they're a virulence factor. Quorum sensing does not help bacteria evade defenses, but it is an element of virulence because bacteria need to cooperate together in order to overwhelm a massive host such as yourself. So if they can't communicate with each other, they can't cooperate and they couldn't overwhelm your body. So glycocalyx is a virulence factor because it helps evade the host's immune system, but it's also a virulence factor because in the form of a biofilm, it makes those chemical conversations more efficient so that the bacteria can cooperate with each other better to overwhelm your body. Antigenic variation. So this one is this is where, information comes in a circular route here. Antigenic variation is very difficult to explain without covering the immune system in more detail. So I have to give you a preview of the immune system before I can explain antigenic variation. So so at the top of the slide here, you have pathogens. So here's a pathogen, here's a pathogen, here's a pathogen. And these are, so let's start with pathogen in red. These red squares on the surface of the pathogen, those are molecules that your body can use to recognize it. Your immune cells learn to recognize pathogens, but not by sight, right? They don't have eyes and there's no light in your body anyway. So the way your immune cells are going to recognize pathogens is through touch. So these shapes on the surface of the pathogen are what your immune cells are going to use to recognize the pathogen. They're going to blindly feel them. I mean, literally, they're going to feel them so they can see what the shapes are what they recognize. So here is supposed to be, so we call them antigens. These molecules on the surface of pathogens, we call them antigens. So you have antigenic variation, where you can already see that we're going to talk about varying these antigens. So here, these are supposed to represent antibodies. Antibodies are receptors that your immune system makes to bind to the antigens on pathogens. So these are from your immune system. They help you recognize the pathogen. Okay. So your immune system makes this. When an antibody binds to a pathogen, it attacks the pathogen. We'll just simplify it and say that. So this antibody, if it binds, it attacks. So when your body the first time this pathogen enters your body, you don't have any of those antibodies. Your body has to generate them slowly in response. So you have this pathogen enters your body, and the pathogen is replicating. That means its antigen numbers are going up. If you were to measure the number of antigens, they're going to go up because the number of pathogens is going up. And there's a little bit of a lag between pathogen growth and the mounting of your immune response. So as the pathogen becomes more numerous, time passes, your immune system is learning about this pathogen, creating the antibodies to attack it. You can see they fit like a lock and key. But then, so and then once you mount your immune response, you start fighting that microbe effectively. So once you start making enough of these antibodies that bind to this antigen, you're killing this pathogen numbers decline. Now, if this were a normal pathogen, that would be the end of it, and your disease would be cleared. But for pathogens that are capable of antigenic variation, what they do is they put on a new antigen. So they change the proteins on their surface. So this is the same pathogen right here, same pathogen. But what it's done is it's put on a mask. So your immune system can no longer recognize it. It's lost the antigens that your immune system got used to. It's put on brand new antigens that your immune system has never seen before. Now your immune system has to start from scratch. It has to build a new set of antibodies that can bind with the new set of antigens on the pathogen. So that's going to take some time. So meanwhile, this pathogen is growing, becoming more and more numerous. Your new immune response to this new antigen is growing. It becomes successful. Your pathogen starts to decline. Now this pathogen switches out its proteins on its surface again. It's put on a new mask. Your immune system now has to start from scratch again, freshly synthesizing a whole new set of antibodies to attack this new antigen on the same pathogen. And this little black antigen here, so this is to show you, of course, pathogens are complicated. They have more than one protein on their surface. So you're making more than one protein at a time. And you're making more than one kind of antibody at a time to fight the same pathogen, I should say. So some of the proteins on the surface of the pathogen might be capable of varying through antigenic variation, and some of them may not be capable of that. So it just depends, it depends on the pathogen. So let me give you some examples. So you can think of antigenic variation as so antigens are the face of the pathogen. When a pathogen is capable of antigenic variation, it means it's able to put on a new face. So you can think of it as putting on a new mask that has to be now learned fresh by your immune system, so your immune system can recognize each new mask in turn. Or if you've ever seen Return to Oz, I really liked Return to Oz. It's very dark if you haven't seen it, but there's the witch Mombi. She collects heads of beautiful women. She's got this whole hallway of heads, and she just puts on a different head for different occasions. So you can think of it as doing that. It's putting on a new face every time, and so it looks different to your immune system. Your immune system has to start from scratch every time it does that. So let me give you some examples of pathogens that you have heard of that are capable of evading your immune response through antigenic variation. You have Neisseria meningitidis, which causes meningitis, and Neisseria gonorrhea, which causes gonorrhea. These two pathogens, so meningitis pili are important in adhesion, but they also are made out of antigens. So they're antigens for which our body responds to with an aggressive immune response. So the pili on meningitis bacteria, our bodies produce antibodies to fight them, and our body produces antibodies aggressively against them. But those same proteins on those pili can be altered. They can be switched out. So Neisseria meningitidis can alter the proteins that comprise the pili so that our immune system response is useless next time around. And then gonorrhea is in the same, it's in the same genus. It can do the same kind of thing. So there's several different kinds of surface proteins that are altered in gonorrhea. There's more than one. And then one more example is actually a protist, not a bacteria. Trypanosoma brucei. This is a protist that causes sleeping sickness. And it actually has thousands of antigen genes that it can cycle through to prevent detection by antibodies. M proteins. So M proteins are surface proteins that you only find on Streptococcus species. So it's just in this genus. M proteins adhere to epithelial cells. Okay. So epithelial cells are surface cells. They can be skin cells or they can be cells on the surface of organs. So they're the outer layer of cells of something. M proteins can also evade phagocytosis. And they can also interfere with the complement system. So there is no way for me to explain the complement system now. I can't explain it until the very end of the immune system chapters. So for now, all you need to know is the complement system is an important piece of our immune system. There's many different immune pathways. It's one immune system pathway. It's actually an immune pathway that, so this immune system pathway initiates inflammation and fever, which actually, I know we don't like inflammation and fever, but actually they're essential elements to fighting infection. So inflammation and fever helps you fight infection. That's part of the complement system. Complement system also helps to destroy pathogenic cells in a variety of ways. So we won't go into detail here, but M proteins interfere with this complement system. It makes it dysfunctional, basically. All right, so M proteins, just on Streptococcus species, they're used as an adhesin, so they're an adhesin, so that's one way it's a virulence factor. They also are difficult to phagocytize, and they interfere with the complement system, which is one critical element of our immune system. Entering a host cell. So a pathogen might hide from your immune cells, your phagocytes, inside one of your cells. All viruses do this, right? All viruses enter the host cell and in order to use transcription translation equipment to replicate, right? And then they burst out of the immune cell or burst out of the host cell or bud out of the host cell in order to go infect another cell. So all viruses are intracellular parasites. And of course, some viruses even become part of your DNA. And that really makes it difficult for your immune system to handle it. In fact, it's impossible then for your immune system to get rid of it. Some bacteria are also intracellular parasites. Intra means within. So intracellular parasites are inside cell parasites. So one example would be tuberculosis. Tuberculosis can live inside your macrophages, which are white blood cells, and replicate inside of them. So we talked in this slide, we talked about antigenic variation, how your white blood cells produce these receptors that bind to antigens on the surface of a pathogen. Well, if the pathogen is inside one of your cells, how are your immune cells going to make those receptors for them? How are the receptors going to bind to them, right? Because they're inside one of your normal human cells. Now, all that your white blood cells can see are normal human cells. They can't see the, quote unquote, see the pathogen anymore. All right, enzymes. There's just one set of enzymes I want to put here in evading host defenses, and that's going to be coagulases. So coagulases cause clotting, blood clotting. And so let me tell you a little bit about blood clotting before I talk about coagulases. So in your body, you have coagulases that help prevent you from bleeding out when you are wounded. So you have all of this in your bloodstream. You have this protein called fibrinogen, and it's dissolved. So you don't see it. It's dissolved in your bloodstream. But when you're wounded, the damaged cells produce a signal that causes this fibrinogen to become converted into fibrin. So what that means is all the little pieces of fibrinogen are linking up together to form these long chains and networks, and that's fibrin. Fibrin is just long chains and networks of fibrinogen. And that netting catches platelets and stops the bleeding temporarily until the tissue is repaired. Okay? So Coagulases are what initiate the fibrinogen to fibrin conversion. So fibrinogen are small proteins dissolved in the bloodstream. Coagulases cause them to convert into fibrin, which is where they all link up to form these networks. And then we call those networks a clot. So it causes the blood to coagulate. So the network catches blood cells, catches platelets, and causes a clot. So when we use coagulases, it's to stop bleeding. Pathogens can come with coagulases. Pathogens can produce some pathogens can produce coagulases as well. And what happens when a pathogenic bacteria produces coagulase is it causes your blood to clot around the pathogen. That's how they build a wall around themselves. So once they have this wall of blood clots around them, your white blood cells can't get to them. They can't get through that wall of blood clot. So in this picture, you have Staphylococcus aureus forming a fibrin capsule that protects it from the immune system of cattle. So the cattle's immune system cannot get it. So I think that some students get confused about coagulases because coagulases are good when we produce them, because we produce them at the right time. We produce them to stop us from bleeding from a wound. But when a pathogen produces them, it's causing a blood clot in the wrong place. It's causing a blood clot around the pathogen that protects the pathogen from our immune system.